Okay, welcome to the review for Chapter 24, which is on speciation. What we're going to be talking about is a few different mechanisms on how new species can arise from different selective pressures. So this diagram here is a really good diagram in your book. It really goes through each step on what barriers two different species have to overcome to create a zygote, which is a hybrid species, and then also have it be viable and fertile, which is a tough one to get to. So a few of the things that separate two different species from actually making a hybrid are these 5 barriers right here. Pre-zygotic refers to before you actually make a zygote or fertilize an egg. So temporal isolation has to deal with time. That deals with if the two different species are going through a mating season at different times of the year, then they won't actually come in contact with each other to create a zygote. Habitat isolation is place. So if the two different species are in completely different areas or in different maybe sections of the same geographic area, they won't actually come in contact with each other. So in doing that, they won't be able to mate and create a zygote. Behavioral isolation is what you do. Behavioral isolation refers to the mating behaviors of some species and how they attract mates. So if you have some bird of paradise dancing around another bird that has no idea what that means, it might be scared and run off. So behavioral isolation could definitely separate two different species from mating. Mechanical isolation is what you have. The parts of the body or parts of the organism that are used for reproduction may not fit together perfectly, and if they don't, they will not be able to actually come together and fertilize and create a zygote. Last here is gamete isolation, and that deals with egg and sperm. Believe it or not, egg and sperm for each species is different sizes and different types. And if the sperm and egg don't actually fit together when two different species mate, then they won't be able to make a zygote in the first place. So let's say we overcome all that, those two species overcome all of those barriers, actually create a zygote. They still have a few more barriers to get over before the offspring is viable and fertile. So one, it reduced viability. That just means that the organism is not sickly and it can actually hunt and fend for itself. So many offspring many zygotes when fertilized from two different species will be actually kind of break down as soon as they're born, or they won't be able to actually fend for themselves, and if that happens, they won't be viable in the next generation. Fertility, reduced hybrid fertility, deals with actually mating and being able to have your own offspring. The example we talked about in class with that were mules. That's the mating between a horse and a donkey, and a horse and a donkey mate, they create a mule, and mules can't actually mate with other mules to create more mules. That never happens. So with reduced fertility, we don't have fertile offspring. Last bit here is hybrid breakdown, and hybrid breakdown refers to you have your offspring, it's viable, it can mate, however the zygotes it actually fertilizes, the zygotes that actually helps create, don't actually make it to completion. They don't actually get born. So with hybrid breakdown, we're talking about the kids of the hybrid that don't actually come to the next generation, and that ultimately stops the new hybrid species from continuing on. So all of those barriers are really the big hurdles that two species that come together to make a hybrid have to get over before that new offspring can be viable, which is lively, and fertile, which is being able to create more offspring. Next thing we're going to talk about are the two ways that you can have speciation. The first way we talked about in class was with allopatric speciation. And with allopatric speciation, what we're looking at is a large population of individuals who are in one particular area, and through some either catastrophe or environmental change, in this example, it looks like a river has divided the population in half. what happens is organisms get separated into two different areas of maybe the same habitat, but now they're in two different populations. Those two new populations will then go through or experience drift because the population size has decreased. So in this example, you see this group of beetles here, which is all in one large population. They get separated by this river, and the very light colored beetle is only going to be on one side. What does that do over generations? Well, you can see in the end here that the lighter beetles happen to be separated on one side of the river compared to the other side, and that's just because that allele made it over to one side of the gene pool and stayed over there. And since drift was happening and the smaller gene pool was on this bottom portion, most of the beetles actually developed into lighter colored beetles. So allopatric speciation has to deal with different place. So if allopatric is different place, sympatric, and actually we'll come back to this picture, sympatric deals with the same place. So we got same here and place. So that's an easy way to remember the two. And with sympatric speciation, what happens is the two different organisms or the two different populations are in the same place, but being in the same place doesn't mean they can automatically just fertilize and create hybrid offspring because they have different genes. Okay, so what happened with this example here? This is what's from your book known as the evolution of bread wheat. And what happened was we had two different species of wheat actually fertilize and create a sterile hybrid. Okay, so that sterile hybrid, which normally would not be able to divide and go through meiosis, actually went through autopolyploidy sympatric speciation. So what happened was these two alleles right here, or these two sets of chromosomes, duplicated, creating an extra set, and through that mitotic error of self-fertilization, we were left with the first type of wheat in this story, which was emmer wheat. So that sympatric autopolyploidy speciation happened in this example right here, because what we're doing is doubling the amount of chromosomes, going from 14 to 28. What happened in the next mitotic error is that emmer wheat fertilized with another type of wheat, creating another sterile hybrid. But through another set of mitotic errors and autopolyploidy speciation, we have another duplication of those chromosomes. So we go from having 28 again now to 42. So we keep doubling the amount of chromosomes in every generation, but through those errors, we are actually creating new species. And that's how we ended up with bread wheat in the end that has enough grain in it that we can actually harvest that and make bread out of. This other example here is kind of referring to parapatric speciation. We talked about that, actually it's with an A, parapatric speciation. And with parapatric, that referred to organisms being in the same place, but a new niche or a new job opening up in that same habitat. So in this example, we have fruit flies that were normally feeding off hawthorns, and these hawthorns were everywhere in the habitat. So normal flies, fruit flies, bred and like mated on those hawthorns. But introducing apples to the situation, introducing apples to the habitat, allowed for another set of flies to start actually mating and feeding on the apples. So what we were left with is, at the start, a lot of flies were just mating and feeding off hawthorns. And after apples were introduced to the population, what happened was the flies that were more suited to breed and feed off of apples, which are over here, actually went through a sort of diversifying selection, where the hawthorn fruit flies were still feeding off of hawthorns, but we also had a large population now feeding off of apples. And through that creation of a new niche, which is the introduction of apples, you can actually have the diversifying effect of creating another species of fruit flies. So that's just another way to divide species and actually go through a speciation event. Next thing we're going to talk about are Hox genes. I know we've been mentioning that a few times throughout the year so far, but it really is a great thing to discuss when talking about speciation, because what evolution really acts on is sets of genes that organize what the body looks like. So Hox genes are really responsible for telling the body where the head goes, where the tail goes, where the midsection of the body is, and all of those genes are present in almost every organism. Okay, and you can see, comparing the mouse and the fruit fly here, they have the same set of Hox genes, roughly, and they have almost the same type of body plan, where you have the head at the front and the tail or abdomen at the back. And with having these Hox genes, what evolution can do to actually create new species here is reorganize those Hox genes and really package bundles of genes in different ways to not only create things like wings and extra legs, but tails and fur. So these different sets of Hox genes can really be shuffled around in order to create new organisms in the end. Last thing we talked about in the chapter was allometric growth. And allometric growth dealt with looking at species that really went through a timing event, a development event, where in this example we can see the skulls of chimpanzees and the skulls of humans. And what happens in the embryos of these two species is that the embryos look roughly the same initially, but through a longer development of the cranium, the skull of a human actually gets a lot larger than the skull of a chimpanzee, and that leads to larger brain size and roughly what led to the separation between chimpanzees and humans. So allometric growth is really looking at the development of organisms and separating those into two different species. Last thing we'll talk about are pediomorphs, and if I can spell this right, that's right. The pediomorphs really look at, again, the development of new species through the timing of when things develop in an individual, but with pediomorphs we're looking at sexual development. So in this salamander, what occurred was this new species of salamander actually developed through the early development of sexual genes. So the sexual, the juvenile, which could not mate, actually developed sexual characteristics early on and kept some of its juvenile look physically. So that thus created a new species of salamander. Same thing can happen with these miniature dogs celebrities carry around in their purse. They can develop sexual characteristics early on, but then keep their juvenile physical characteristics, keeping them small and cuddly. So that's really the review for Chapter 24. If you have any other questions, feel free to ask your teacher, and we'll see you next time.